Chapter 22, Metabolism and Energy Balance. Let's discuss the appetite and satiety. This slide introduces the concept of appetite and satiety, which plays a vital role in regulating food intake, a complex and highly controlled process. Here's the breakdown. There are two competing behavioral states, appetite and satiety. Appetite, or hunger, drives the need to consume food. It is the body's signal that energy reserves may need replenishment. Satiety is feeling of fullness or satisfaction that suppresses further eating. It ensures the body does not overconsume. The hypothalamus acts as the central command center for regulating these states, containing two key control centers. Feeding center stimulates hunger, encouraging food consumption. Satiety center suppresses hunger, promoting the feeling of fullness and signaling that energy needs have been met. This dynamic interplay between appetite and satiety ensures energy homeostasis in the body. It's a finely tuned system influenced by hormones, neural signals, and even psychological factors. This slide focuses on energy balance, which involves accounting for all the energy that enters the body through food. The body uses this energy in different ways, primarily through chemical reactions that either convert it into heat or harness it to perform work. There are three categories of cellular work. Transport work moves molecules or ions across cell membranes, such as sodium-potassium pumps that help maintain cellular function. Mechanical work includes actions like muscle contraction and organ movement. Chemical work encompasses synthesis and storage of energy. Short-term storage, as ATP, which cells use immediately. Long-term storage, as glycogen or fat, used when energy is needed over extended periods. Most of the work done by cells, such as maintaining homeostasis, occurs unconsciously. However, we can consciously influence energy balance. Increasing energy output, for example, through physical activity, or decreasing energy intake, such as by eating less. Energy balance is critical to maintaining the body's functionality and overall health, ensuring that input from food matches output through work and heat. Metabolism is the body's engine, constantly burning fuel to sustain life. At its core is the basal metabolic rate, BMR, the minimum energy required to keep us alive, measured after a 12-hour fast when the body is at complete rest. A close cousin, the resting metabolic rate, gives us insight into energy expenditure under slightly less strict conditions. But what factors shape this metabolic rate? There are 6 key players. With age, metabolism slows, and between sexes, men typically burn more due to higher lean muscle mass. Speaking of muscle, it's an energy powerhouse, requiring more fuel even when at rest compared to fat. Movement, whether a brisk walk or intense workout, revs the metabolic engine. What we eat and how our bodies process it plays a vital role. Some foods even spark thermogenesis, a calorie-burning fire during digestion. The body's chemical messengers, like thyroid hormones, can act as accelerators or brakes on metabolism. Inherited traits lay the foundation, determining our metabolic blueprint. Here's the empowering part. While some factors, like age or genetics, are beyond our control, we hold the reins to others. By consciously adjusting energy intake and increasing physical activity, we can directly influence our metabolic rate. And for those seeking to supercharge this system, strength training is the secret weapon. Building lean muscle transforms the body into a calorie-burning machine, using energy even at rest while reducing fat storage. In this dynamic dance of energy, the choices we make shape the metabolic music we play. Our bodies are like well-designed storage systems, holding energy in 2 main forms, glycogen and fat. Let's take a closer look at these 2 vital energy reserves. Glycogen, a glucose polymer, is our body's immediate energy currency. It is primarily stored in the liver and skeletal muscles, ready to release glucose when quick energy is needed. While efficient for short-term energy demands, glycogen reserves are limited, making it a fast but temporary solution. Fat, on the other hand, is the master of compact energy storage. With more than twice the energy content of an equal amount of carbohydrates or protein, fat is the go-to source for long-term energy needs. However, this efficiency comes with a trade-off. Energy stored in fat is harder and slower to access, requiring more effort for the body to break down and utilize. To put it in perspective, proteins and carbohydrates provide 4 kcal per gram. Fats, however, pack a whopping 9 kcal per gram. Together, glycogen and fat ensure that our bodies have a balance between quick energy and long-lasting reserves, powering us through both immediate and prolonged demands. Whether sprinting through a task or enduring a marathon, these energy stores work in harmony to sustain life. Metabolism is the orchestra of life, with 2 main pathways acting as its fundamental players: anabolic pathways and catabolic pathways. Let's dive into how they maintain balance in our bodies. In the anabolic realm, smaller molecules come together to create larger, more complex ones. Think of it as the body's construction process, synthesizing essential components like proteins, fats, and glycogen. This occurs during the fed state, also known as the absorptive state, when nutrients are abundant and the body focuses on storage and growth. On the flip side, catabolic pathways deconstruct large molecules, breaking them down into smaller, energy-releasing units. This process is essential during the fasted state, or the post-absorptive state, when stored resources are tapped to fuel the body's activities. Together, these 2 metabolic pathways work in harmony, ensuring the body can build when resources are plentiful and adapt when they are scarce. When we consume food, the energy derived from ingested biomolecules is not simply used up, but has 3 potential fates that ensure our bodies function efficiently and prepare for future needs. There are 3 fates of ingested biomolecules. The first is energy for mechanical work. Some of the biomolecules are converted directly into energy to power physical activities, from cellular functions to muscle contractions. The second is synthesis for growth and maintenance. A portion is directed toward building and repairing the body's tissues, supporting growth, and maintaining vital structures. The third is storage as glycogen or fat. Any excess energy is stored, either as glycogen in the liver and muscles or as fat in adipose tissues, serving as a reservoir for future energy needs. Nutrient pools for immediate use in plasma as either fatty acid or glucose. In the plasma, nutrient pools are constantly available to meet the body's energy demands. Free fatty acids pool can be used as a rich energy source. Glucose pools are tightly regulated because the brain relies heavily on glucose for energy. Processes like glycogenesis, or synthesis of glycogen, and lipogenesis, or synthesis of fat, glycogenolysis, or breakdown of glycogen, and gluconeogenesis, or generation of glucose from non-carbohydrate sources. Amino acid pool are used for protein synthesis and, when necessary, as an alternative energy source. This intricate system ensures the body is well prepared to balance immediate energy requirements while safeguarding reserves for future demands. Here, we explore the dynamic regulatory mechanism in metabolism, where enzymes catalyze opposing reactions to ensure a balance between synthesis and breakdown of molecules. Let's break this down. Panel A. Without regulation. In the absence of regulation, 2 enzymes catalyze forward and reverse reactions between substrates A and B. This results in a back-and-forth cycle with no net production of either substrate. It's an inefficient state of equilibrium. Panel B. Fed state metabolism. Glycogen synthesis. In the fed state, insulin dominates. Insulin promotes enzyme activity for glycogen synthesis from glucose while inhibiting enzymes responsible for glycogen breakdown. The result is a net synthesis of glycogen, as the body stores energy from the ingested nutrients. Panel C. Fasted state metabolism. Glucose synthesis. In the fasted state, glucagon takes over. Glucagon activates enzymes that break down glycogen into glucose while suppressing enzymes for glycogen synthesis. This leads to net synthesis of glucose, ensuring the body has energy when external nutrients are unavailable. This push-pull control allows the body to dynamically shift metabolic processes to meet the changing energy demands of fed and fasted states, maintaining homeostasis. In fed-state metabolism, we delve into how the body processes nutrients right after a meal when energy is abundant. Carbohydrates are metabolized to produce ATP, the energy currency of the cell. Any excess glucose is stored. Glycogen serves as short-term energy storage, primarily in the liver and muscles. Surplus glucose is converted to fat, providing long-term energy reserves. Amino acids are essential for synthesizing proteins, which are crucial for structural components, such as muscle and connective tissue. Functional roles, like enzymes, hormones, and neurotransmitters. If there's an excess of amino acids, they are either burned as an energy source or converted into fat for storage. This phase ensures the body efficiently stores and utilizes nutrients to maintain energy balance and prepare for future energy demands. In the Fats Store Energy slide, we explore how fats play a crucial role in energy storage and utilization. Chylomicrons, or lipid-rich particles, exit the intestines through lymphatic vessels. They contain a mix of cholesterol, triglycerides, phospholipids, and apoproteins, essential components for energy and cellular functions. Lipoprotein lipase, an enzyme, breaks down triglycerides into glycerol and fatty acids, making them accessible for cellular use or storage. As for cholesterol regulation, excess cholesterol doesn't go to waste. It is secreted into bile, aiding digestion, or into the bloodstream as part of lipoprotein complexes, ensuring balance and transport within the body. In the fed state, the body synthesizes lipids, even from non-lipid precursors, ensuring ample energy storage for future use. This process demonstrates the efficiency of the body in managing fats, utilizing them as an immediate fuel source or storing them for long-term energy demands. This figure discusses fat synthesis. Here, it outlines the journey of dietary fats through the body, demonstrating their digestion, absorption, transport, and utilization. Step 1. Breakdown of dietary fats. Bile salts, secreted from the liver, emulsify dietary fats in the intestinal lumen, breaking them down into monoglycerides, free fatty acids, and cholesterol. Step 2. Formation of chylomicrons. Inside the intestinal epithelial cells, these components are reassembled into chylomicrons, large lipoprotein particles that carry dietary fats along with phospholipids and apoproteins. Step 3. Transportation via lymphatic system. Chylomicrons are transported into the lymphatic vessels, bypassing the liver initially, and then into the bloodstream. Step 4. Lipoprotein lipase action. In the blood, lipoprotein lipase breaks down triglycerides in chylomicrons into glycerol and free fatty acids, making them available for uptake by cells. Step 5. Adipose storage or cellular use. Adipose cells reassemble these free fatty acids and glycerol into triglycerides, storing them as long-term energy reserves. In other cells, free fatty acids are oxidized for immediate energy production. Step 6. Chylomicron remnants and HDLC. The remaining chylomicron particles and high-density lipoprotein cholesterol return to the liver for further processing. In the liver, low-density lipoproteins and very low-density lipoproteins are synthesized for cholesterol transport or recycled into bile salts. Step 7. Cholesterol distribution. LDLC travels through the bloodstream to most cells, where cholesterol is used for membrane synthesis or hormone production. This system highlights the efficiency of fat metabolism, ensuring the body can store excess energy, produce immediate fuel, and regulate cholesterol balance. This diagram summarizes the interconnected biochemical pathways responsible for energy production in the body. The process begins in the cytoplasm with glucose, glycerol, and amino acids. Glucose, stored as glycogen in the liver and muscles, is converted into glucose 6-phosphate and enters glycolysis. Glycolysis breaks down glucose to form pyruvate while producing a small amount of ATP. Pyruvate serves as a critical branching point. Under anaerobic conditions, pyruvate is converted into lactate. In aerobic conditions, pyruvate enters the mitochondria and is converted into acetyl-CoA. Acetyl-CoA is a central molecule in metabolism. It feeds into the citric acid cycle, generating high-energy electrons in the form of NADH and FADH2. These electrons are transported to the electron transport system, where they generate ATP, the main energy currency of cells, through oxidative phosphorylation. This pathway also produces water and carbon dioxide as byproducts. Additionally, acetyl-CoA can contribute to the synthesis of fatty acids or ketone bodies in the liver, providing alternative energy sources during fasting or low glucose availability. Some amino acids and glycerol also integrate into these pathways, illustrating the versatility of metabolic systems to meet the body's energy demands. Let's explore how the body processes nutrients during times of abundance and scarcity. In the fed state metabolism, when food is readily available, the body prioritizes energy storage and synthesis. Carbohydrates, absorbed as glucose, fructose, and galactose, become the star players. Glucose is quickly used for energy through glycolysis in the citric acid cycle, stored as glycogen in the liver and muscles, or converted into fat for long-term energy storage. Proteins, absorbed as amino acids and small peptides, primarily go toward building structural and functional proteins. However, when there's an excess, amino acids are converted into fat and stored, ensuring nothing goes to waste. Meanwhile, fats, absorbed as fatty acids, triglycerides, and cholesterol, are efficiently stored in adipose tissue or used for important roles like membrane synthesis and hormone production. But in the fasted state metabolism, when food is scarce, the body switches to survival mode. Glycogen, the stored form of glucose, is broken down to fuel vital processes, and glucose from this breakdown powers the brain and body. Proteins become a secondary source, with amino acids deaminated to create energy or converted to glucose through gluconeogenesis. Fats play a key role, with triglycerides breaking down into fatty acids and glycerol, which are further oxidized to provide a steady stream of ATP. Through these elegant pathways, the body seamlessly transitions between energy storage and utilization, maintaining balance and ensuring survival in both feast and famine. Fat synthesis is a vital metabolic process that converts excess glucose into stored energy. It all begins with glucose, which is broken down through glycolysis to form glycerol. This glycerol serves as the backbone for triglyceride synthesis. Next, fatty acids are crafted. This occurs when two carbon acyl units derived from acetyl-CoA are linked together by fatty acid synthetase. These acyl units, building blocks of fatty acids, are essential for constructing the long hydrocarbon chains that form fat. Finally, these components come together. One molecule of glycerol combines with 3 fatty acids to create a triglyceride, the primary form of fat stored in adipose tissues. Additionally, some acetyl-CoA can be redirected for cholesterol synthesis, further showcasing the flexibility of this metabolic pathway. Through this process, the body ensures energy is efficiently stored for future use, illustrating its remarkable ability to adapt to varying energy demands. Cholesterol plays a pivotal role in our body, but its distribution and levels are key indicators of heart health. LDL-C, commonly referred to as bad cholesterol, carries cholesterol from the liver to most cells. However, only the small and dense forms of LDL are considered lethal cholesterol due to their strong association with atherosclerosis. On the other hand, HDL-C, known as good cholesterol, works to remove cholesterol from the plasma, effectively transporting it back to the liver or other pathways for processing. Abnormal plasma lipid levels, particularly elevated LDL-C or decreased HDL-C, significantly increase the risk of atherosclerosis and coronary heart disease. These imbalances in cholesterol levels highlight the importance of monitoring lipid profiles for cardiovascular health. To address these risks, therapeutic drugs are often designed to target cholesterol absorption and metabolism, offering a way to mitigate the potential damage caused by abnormal cholesterol levels. In the fasted state, the body shifts its focus to maintaining energy levels by utilizing stored resources. Glycogen, stored in the liver and muscles, is converted back into glucose through the process of glycogenolysis to maintain blood glucose levels. Additionally, when glycogen stores are depleted, the body activates gluconeogenesis, a process that forms glucose from non-carbohydrate sources like amino acids and glycerol. Proteins can also contribute to energy production. Through deamination, amino acids are stripped of their nitrogen group, producing ammonia as a byproduct. Ammonia is then converted to urea, which can be measured in a blood test as blood urea nitrogen, or BUN. Lipids, being the most energy-dense macromolecules, play a crucial role in the fasted state. Through lipolysis, triglycerides are broken down into glycerol and free fatty acids. Glycerol enters glycolysis, while fatty acids undergo beta-oxidation to form acetyl-CoA. However, when there is an excess of acetyl-CoA, it can be converted into ketone bodies, a backup energy source for the brain and muscles during prolonged fasting or starvation. Yet, this process carries risks. High levels of ketone bodies can lead to ketoacidosis, a dangerous condition characterized by acidic blood pH. This metabolic state highlights the body's remarkable ability to adapt and prioritize survival in the absence of food. In amino acid catabolism, the body breaks down amino acids to harness energy or build other molecules. This process begins with deamination, where the amino group is removed from the amino acid. This step produces an organic acid, which can enter metabolic pathways like glycolysis or the citric acid cycle for energy production. During deamination, ammonia, NH3, is released as a byproduct, which is highly toxic if allowed to accumulate. To protect the body, ammonia undergoes a conversion process. It combines with a hydrogen ion to form ammonium, which is then transformed into urea in the liver. This detoxified urea is safely excreted through the urine. The urea production is reflected in blood tests as blood urea nitrogen, or BUN, a measure of kidney and liver function. This cycle underscores the body's ability to utilize proteins as an energy source during fasting or when carbohydrate and fat stores are insufficient, while carefully managing potentially harmful byproducts. Gluconeogenesis is the metabolic process by which the body generates glucose from non-carbohydrate sources, ensuring a steady supply of glucose, especially during fasting or intense exercise. This process predominantly occurs in the liver and, to a lesser extent, in the kidneys. The pathway starts with precursors like amino acids, lactate, and glycerol. Lactate, produced from anaerobic glycolysis in muscles, and amino acids, derived from protein breakdown, are converted into pyruvate. Glycerol, originating from fat breakdown, feeds into the process further upstream. Through a series of enzymatic reactions, these molecules are transformed into glucose-6-phosphate and ultimately into glucose. This newly synthesized glucose can be released into the bloodstream to maintain blood sugar levels or stored as glycogen. Gluconeogenesis highlights the body's remarkable adaptability in maintaining energy balance and supporting critical tissues like the brain and red blood cells when dietary carbohydrates are unavailable. Lipolysis is the metabolic process that breaks down triglycerides stored in fat cells into usable energy sources. This process begins when lipase enzymes act on triglycerides, splitting them into glycerol and 3 fatty acids. The glycerol component enters the glycolysis pathway, where it is converted into intermediates that feed into energy production cycles like the citric acid cycle. Meanwhile, fatty acids undergo a separate pathway known as beta-oxidation. This occurs in the mitochondrial matrix, where each fatty acid is systematically broken down into 2 carbon acyl units. These acyl units are converted into acetyl-CoA, a critical molecule that feeds directly into the citric acid cycle for ATP generation. This dual approach allows the body to efficiently utilize stored fats during periods of energy demand, such as fasting or exercise, highlighting the essential role of lipolysis in maintaining energy balance and cellular function. Metabolism is finely regulated by the interplay between two key hormones secreted by the pancreas, insulin and glucagon. These hormones are produced in the islets of Langerhans, with beta cells responsible for secreting insulin and alpha cells for producing glucagon. The balance between these two hormones, the insulin to glucagon ratio, is the primary regulator of metabolic states in the body. In the fed state, when glucose is abundant after a meal, insulin dominates, promoting glucose uptake and storage as glycogen or fat. In contrast, during the fasting state, when blood glucose levels drop, glucagon dominates, stimulating the breakdown of glycogen and the production of glucose through gluconeogenesis to maintain blood sugar levels. This intricate balance ensures that energy is available to meet the body's needs, regardless of whether it is in a fed or fasting state. The pancreas is a dual-function gland located just behind the stomach, with both exocrine and endocrine roles. This illustration provides a detailed look at its anatomy and cellular structure. On the left, we see the pancreas in its gross anatomy. It's connected to the digestive system through the pancreatic duct, which joins the common bile duct and empties into the small intestine, duodenum. This exocrine portion of the pancreas secretes digestive enzymes into the intestine to aid in digestion. Zooming into the cellular level, the central image highlights the islets of Langerhans, which are clusters of endocrine cells scattered throughout the pancreas. These islets contain beta cells, which secrete insulin and amylin to regulate blood sugar levels, alpha cells, which secrete glucagon, promoting glucose release into the blood, delta cells, which produce somatostatin, a hormone that regulates both insulin and glucagon secretion. To the right, the microscope image, magnified at 350x, shows the actual structure of these islets of Langerhans surrounded by the exocrine tissue. The balance between the functions of these cells plays a critical role in maintaining glucose homeostasis in the body. This figure demonstrates the intricate balance between insulin and glucagon, two critical hormones secreted by the pancreas, which regulate metabolism and maintain blood glucose homeostasis. This figure is divided into three main panels. Panel A. Fed state metabolism, or insulin dominates. In the fed state, insulin takes center stage. Following a meal, the pancreas releases insulin, tipping the metabolic balance towards storage and synthesis. Insulin promotes glucose oxidation to produce energy, glycogen synthesis for short-term energy storage, fat synthesis for long-term storage, protein synthesis, aiding in tissue repair and growth. Panel B. Fasted state metabolism, or glucagon dominates. In contrast, during the fasting state, glucagon becomes the dominant hormone. It helps maintain blood glucose levels by shifting the balance towards catabolic pathways, including Glycogenolysis, breaking down glycogen into glucose, Gluconeogenesis, synthesizing glucose from non-carbohydrate precursors, Ketogenesis, forming ketone bodies from fatty acids as an alternative energy source. Panel C. Graph of glucose, insulin, and glucagon dynamics. This panel visualizes hormone levels before and after a meal. Top graph. Plasma glucose concentration, shown in the dashed line, rises after a meal, triggering an increase in plasma insulin, shown in the red line, to facilitate glucose uptake and storage. Bottom graph. As insulin rises, plasma glucagon concentration, shown in the green line, decreases. In fasting states, the trend reverses. Glucagon levels rise to stimulate glucose production. Overall, this balance between insulin and glucagon ensures that cells have a continuous energy supply, whether in a fed or fasting state. In the fed state, insulin emerges as the dominant hormone, orchestrating the body's storage and utilization of nutrients. This slide highlights the key factors that stimulate insulin secretion. First, we have increased plasma glucose. After a meal, rising blood glucose levels signal the pancreas to release insulin, promoting glucose uptake by cells. Next, we have increased plasma amino acids. Elevated levels of amino acids in the bloodstream further stimulate insulin secretion, aiding in the synthesis of proteins. Now, we have feed-forward effects of GI hormones. Hormones released by the gastrointestinal tract in anticipation of food intake enhance insulin secretion. This prepares the body to manage the incoming nutrient load effectively. Lastly, we have parasympathetic activity. The parasympathetic nervous system, activated during digestion, also plays a role in stimulating insulin release. Through these mechanisms, insulin ensures that the body efficiently stores and utilizes nutrients, maintaining metabolic homeostasis in the fed state. This diagram illustrates the regulatory pathway of insulin secretion during the fed state, showcasing how the body processes and responds to nutrient intake. The journey begins with food consumption, triggering multiple sensory and digestive responses. Sensory receptors and central nervous system (CNS) input detect the presence of food, stimulating parasympathetic activity. As digestion progresses, the gastrointestinal tract detects carbohydrates, proteins, and lipids. The gut releases hormones such as GLP-1 and GIP, which signal pancreatic beta cells to prepare for insulin secretion. Nutrient digestion and absorption lead to elevated glucose and amino acid levels in the bloodstream, further stimulating beta cells. The pancreas releases insulin into the bloodstream. Systemic response to insulin include liver, muscle, adipose, and other cells. In liver, insulin promotes glycogenesis, lipogenesis, and protein synthesis while inhibiting glucose production. In muscle, adipose, and other cells, insulin facilitates glucose uptake and storage by increasing glucose transporters on the cell membrane. Negative feedback loop. As plasma glucose levels decrease, insulin secretion diminishes, maintaining homeostasis. This tightly regulated feedback system ensures that the body efficiently manages nutrient absorption and storage in the fed state. Insulin, a key anabolic hormone, plays a vital role in promoting the storage and utilization of nutrients within the body. Here's how. Insulin binds to tyrosine kinase receptors located on the cell membrane. This activation initiates a cascade of intracellular signaling. Insulin effectively decreases blood sugar levels by increasing glucose transport into cells. Specialized transporters are mobilized to the cell membrane, enabling glucose uptake. Once inside the cells, insulin directs glucose towards pathways of utilization, such as glycolysis for energy production or storage, converting it into glycogen in the liver and muscles. Insulin promotes the incorporation of amino acids into proteins, aiding in protein synthesis necessary for growth and repair of tissues. Beyond proteins and glucose, insulin stimulates the synthesis of lipids, converting excess nutrients into stored fat for long-term energy reserves. In essence, insulin orchestrates a coordinated effort to drive the body into a state of nutrient utilization and storage, ensuring homeostasis and preparation for periods of fasting. Insulin's journey into the cellular world begins with its binding to the tyrosine kinase receptor on the cell membrane. This is the first critical step, as it activates the receptor and sets off a chain reaction inside the cell. Step 1: Binding and activation. Insulin attaches to its receptor, triggering the phosphorylation of insulin receptor substrates, or IRS, inside the cytoplasm. Step 2: Signal transmission. These phosphorylated IRS proteins act as messengers, activating second messenger pathways. These pathways fine-tune cellular activities by altering protein synthesis or modifying existing proteins. Step 3: Cell membrane transport. One key result of these pathways is the modification of membrane transport systems. For example, GLUT4 transporters are moved to the cell membrane, enhancing the cell's ability to take in glucose. Step 4. Metabolic adjustments. With more glucose entering the cell, enzymes are activated or inhibited to direct glucose into storage pathways, such as glycogenesis or utilization processes, like glycolysis. Step 5. Transcriptional influence. These pathways also affect transcription factors in the nucleus, modifying long-term protein production to adapt to the body's metabolic needs. Ultimately, insulin ensures that cells efficiently take up glucose and maintain the delicate balance of energy storage and utilization, highlighting its vital role in metabolic homeostasis. Glucose transport varies significantly between the fasted and fed states, depending on insulin availability and the cell's needs for glucose. On the left panel, we have the fasted state. When insulin is low or absent, the GLUT4 transporters, which is the proteins responsible for glucose uptake, are not present in the cell membrane of adipose, or fat, and resting skeletal muscle cells. As a result, glucose cannot enter the cell, and the cell is not able to take in glucose from the bloodstream efficiently. The cell remains in a state where glucose uptake is minimal, prioritizing the body's need for energy in other areas. On the right panel, we have the fed state. When insulin levels rise following a meal, it binds to the insulin receptor on the cell membrane, triggering a signal transduction cascade inside the cell. This cascade activates a series of processes that prompt the insertion of GLUT4 transporters into the cell membrane through exocytosis. With the transporters now embedded in the membrane, glucose is able to move into the cell, where it can be used for energy production or stored as glycogen for future use. This process is a clear example of how insulin regulates glucose uptake, ensuring that cells can access and utilize the energy from food during the fed state. The liver's role in glucose transport differs between the fasted and fed states, as shown in this illustration. Let's explore the two scenarios. On the left, we have the fasted state. During fasting, insulin levels are low, and the liver is actively producing glucose. Using GLUT2 transporters, glucose is transported out of the hepatocyte into the bloodstream to maintain blood glucose levels. This glucose originates from two main processes: glycogenolysis, or breakdown of glycogen stores, and gluconeogenesis, or synthesis of glucose from non-carbohydrate sources. Here, the hepatocyte ensures a steady glucose supply to fuel other tissues during periods of fasting or low dietary intake. On the right, we have the fed state. After a meal, insulin levels rise, triggering a signal cascade in hepatocytes. This cascade reverses the glucose concentration gradient. Now, glucose flows into the hepatocyte through GLUT2 transporters. Once inside, glucose is rapidly converted to glucose 6-phosphate by hexokinase, keeping the intracellular glucose concentration low and enabling continued glucose uptake. This glucose can then be stored as glycogen or utilized for energy production, supporting the liver's role in nutrient storage and metabolism. This dynamic regulation highlights the liver's adaptability, ensuring blood glucose homeostasis during both nutrient abundance and scarcity. In the fasted state, glucagon becomes the primary hormone controlling blood glucose levels. Let's break this down. Glucagon is an antagonist to insulin. While insulin promotes glucose uptake and storage during the fed state, glucagon has the opposite role. It works to prevent hypoglycemia, dangerously low blood glucose levels, by increasing the availability of glucose in the bloodstream. Liver as the primary target. Glucagon's main site of action is the liver. Here, it stimulates two critical processes: glycogenolysis, the breakdown of stored glycogen into glucose, which is then released into the blood; gluconeogenesis, the synthesis of new glucose molecules from non-carbohydrate precursors, such as amino acids and glycerol. Trigger for glucagon release is caused low blood glucose levels serve as the key signal for glucagon secretion. This ensures that, even during fasting, the body maintains adequate blood glucose levels to fuel essential organs, such as the brain. In summary, glucagon is the body's safeguard against hypoglycemia, acting predominantly through its effects on the liver, to mobilize and produce glucose. In response to hypoglycemia, or low plasma glucose levels, the body triggers an intricate endocrine mechanism to restore balance. The decline in plasma glucose activates alpha cells of the pancreas, leading to an increase in glucagon secretion. Simultaneously, reduced plasma glucose suppresses beta cells of the pancreas, decreasing insulin secretion. Glucagon targets the liver, initiating processes to restore blood glucose. Glycogenolysis is where stored glycogen is broken down into glucose. Gluconeogenesis is where glucose is synthesized from non-carbohydrate precursors like lactate, pyruvate, and amino acids. Ketogenesis occurs in prolonged hypoglycemia, where fatty acids are converted into ketone bodies, an alternative energy source for the brain and peripheral tissues. With reduced insulin, glucose uptake by muscle, adipose tissue, and other cells is minimized, preserving glucose for essential organs like the brain. As plasma glucose rises, a negative feedback mechanism inhibits further glucagon release, maintaining glucose levels within normal range. This intricate system underscores the body's ability to prioritize critical energy needs during hypoglycemia, ensuring glucose is available for vital functions, particularly for the brain. Diabetes mellitus, commonly referred to as DM, represents a group of metabolic disorders characterized by abnormally elevated plasma glucose levels, a condition known as hyperglycemia. The persistent rise in blood sugar can lead to severe complications over time, affecting blood vessels, the eyes, kidneys, and the nervous system. The two primary types of diabetes mellitus are: Type 1 diabetes mellitus results from an insulin deficiency caused by the autoimmune destruction of beta cells in the pancreas. Without sufficient insulin, glucose cannot enter cells effectively, leading to elevated blood sugar levels. Type 2 diabetes mellitus is where the body becomes insulin resistant, meaning that even though insulin is present, the cells do not respond effectively. This condition is often associated with lifestyle factors, genetics, and obesity. Both types of diabetes highlight the critical role of insulin in regulating blood glucose levels, the potential systemic effects of impaired glucose control. Managing diabetes effectively is essential to prevent complications and maintain overall health. Type 1 diabetes mellitus, DM, often emerges in childhood, influenced by a genetic predisposition and sometimes triggered by a viral infection. In individuals with type 1 DM, the absence of insulin results in the body entering a fasted state metabolism, creating profound metabolic disruptions. Here's what occurs in the absence of insulin. Protein metabolism is where the body resorts to breaking down proteins, as cells cannot utilize glucose effectively for energy. Fat metabolism is where fat breakdown accelerates, producing ketone bodies as an alternative energy source, contributing to metabolic imbalances. In glucose metabolism, without insulin, blood sugar levels rise dramatically, leading to hyperglycemia. In brain metabolism, the brain perceives energy starvation despite high blood glucose levels, prompting excessive hunger and eating, a condition known as polyphagia. Osmotic diuresis and polyuria occurs due to high glucose levels spill into the urine, or glucosuria, drawing water with it. This leads to excessive urination, or polyuria. Dehydration occurs due to increased urination, prompting excessive thirst and drinking, or polydipsia. Metabolic acidosis occurs due to the breakdown of fats, thus producing ketone bodies, which accumulate in the blood. This condition, called ketoacidosis, can disrupt the body's pH balance and becomes a medical emergency. These metabolic changes highlight the critical role of insulin in maintaining energy balance and preventing life-threatening complications. Proper management and monitoring of type 1 diabetes are essential to avoid these severe outcomes. Type 2 diabetes is the most common form of diabetes, accounting for nearly 90% of all diabetes cases. Unlike type 1 diabetes, where insulin is deficient, individuals with type 2 diabetes often have elevated insulin levels due to a condition known as insulin resistance. This means their body's cells do not respond effectively to insulin, impairing glucose uptake and leading to elevated blood sugar levels. Managing type 2 diabetes typically begins with lifestyle interventions. Exercise and weight loss are the first-line therapies. These strategies improve insulin sensitivity, allowing cells to respond better to the body's insulin. If lifestyle changes fail to control blood sugar levels, drug therapy is introduced as the next step. These medications work to enhance insulin action or lower glucose production in the liver. Early intervention is key to preventing complications and improving quality of life for those living with type 2 diabetes. Metabolic syndrome is a cluster of conditions that significantly increase the risk of both type 2 diabetes and cardiovascular disease. It represents a dangerous combination of obesity, atherosclerosis, and high blood pressure. To diagnose metabolic syndrome, individuals must present with at least 3 of the following criteria. Central obesity is defined by waist circumference exceeding 40 inches in males or 35 inches in females. Elevated blood pressure are readings above 130/85 millimeters Hg. Fasting blood glucose greater than 110 milligrams/dl, indicating insulin resistance. Elevated fasting plasma triglycerides is a marker of disrupted lipid metabolism. Low plasma HDL-C levels, often referred to as the good cholesterol. This condition acts as a bridge, linking the metabolic disturbances seen in diabetes to the development of cardiovascular disease, emphasizing the importance of early intervention to manage and mitigate these risks. To wrap up today's lecture, let's review the key takeaways. We explored the physiological signals that regulate hunger and fullness, emphasizing their role in controlling food intake. We discussed the dynamic balance between energy intake and energy expenditure and its importance in maintaining a healthy metabolic state. We reviewed the biochemical pathways that convert food into energy, highlighting how metabolism supports cellular and systemic functions. We covered the processes that occur in the fed state, when the body prioritizes nutrient storage and utilization following food intake. We examined how the body shifts to relying on stored energy during periods of fasting and the importance of this metabolic flexibility. Finally, we talked about the intricate systems that maintain metabolic homeostasis, ensuring stability despite external and internal changes. These concepts form the foundation for understanding how the body manages energy and nutrients. As you reflect on today's lecture, consider how these processes are interconnected and essential for maintaining health.